Okay, so when we're learning about plants, the first thing we need to know is the structure of the root, the stem, and the leaf, and where we are going to find all the different tissues that we have within that root, stem, and leaf, and to be able to label them or point out where certain things are. So if I start with the root, so you will always have a plant root that looks like this. So you have extensions coming out of it. Then you have multiple layers and you have this X shape in the middle. So starting from the outside and working our way in, our extensions that we have all the way out, those would be your root hairs. And now the root hairs are there to increase the surface area so that we can get more water and minerals and things to get into the root. So those would grow out in between the soil to increase that surface area for water movement. And they would be extensions from the outermost layer, which would be the epidermis. So you're always going to have the outer layer of the epidermis. Then directly below that, okay, that layer in the middle would be all your packing tissue, and that would be in the cortex. And now that cortex is made of parenchyma cells. So those are just dense, large cells that would be packed tightly in there to make up your cortex. Then you would get to the inner layers and you would have the first inner layer, which we call the pericycle, followed by the one directly below it, which would be an endodermis on there. And then below that, you are going to get to this cross-shaped tissue and those tissues in between, which are the important tissues, because that cross shape would always be the xylem. And sitting in the arms of the xylem over there would be the phloem. And your xylem and your phloem are your transport tissues for our plant system. So our xylem is going to transport water and our phloem is going to transport sugars. And in a root, you're always going to have the xylem in the middle in that shape with the phloem in the corners of the arms. Okay, so then if we look at the stem, now the stem looks slightly different because all those roots join together to form that thick stem. So now in the stem, okay, we no longer have any root hairs because we don't need to touch through the soil to increase the surface area. All we have on the outside is going to be the epidermis. Then directly below it, we're still going to have our packing tissue, so that's still going to be called the cortex. But then you have these structures like this going all the way around, okay, and those would be our vascular bundles. And in the vascular bundle, you are going to have tissue on the inside, which is going to be the xylem. Then the tissue on the outside is going to be the phloem. And it's going to be separated in the middle by a different kind of tissue that we call cambium. And all together, they make a vascular bundle. Okay, and there can be any number of those in the plant, depending on how big or small that plant is. And in the very center, we are going to have more packing tissue, which forms what we call the pith. And that is all going to be parenchyma tissue in the cortex and the pith. Again, they're just in different areas. So that's what our stem is going to look like. And then the last one is the structure of the leaf. So now our leaf is going to have layers in there, okay? And we're going to start from the top and work our way down. So at the very top, those cells over there are going to be your epidermis, okay? But since they're at the top, we say it's the upper epidermis. And it usually has a layer on top that we call the cuticle. Now the cuticle is a thick waxy layer that can be there to help prevent water loss. The upper epidermis is a layer of transparent cells to allow sunlight to move through to get to the layer underneath. And that layer underneath would be your palisade mesophyll. And that's where you have all your chloroplasts in there to be able to photosynthesize and absorb that sunlight. Okay, and then in the layer underneath that, over there, you're going to have spongy mesophyll. So the palisade mesophyll is those long, densely packed ones. Spongy mesophyll, there's more air spaces and they're more round. And then in between all of that, you are going to have an area like this, which we call our veins, okay, which would be made up of those vascular bundles, just like in the stem. So this tissue over here at the top would be the xylem. Okay, and we know it's the xylem because they're often irregular shaped with pretty thick walls. Then below that, underneath at the bottom, facing downwards, would be the phloem. And you're going to identify that because you usually have your tubes, but with other cells next to them. And in the middle of that, you would still have your cambium. And then you get to the bottom layer, which we would then call the lower epidermis. And in there where you have your air spaces with two cells on the side, you would call the stomata. So the stomata is made up of the gap in the middle of the stoma with two guard cells on either side for the openings, and that is then for gaseous exchange to occur so that we can get our carbon dioxide that we need for photosynthesis and release oxygen. So that is then the structure of the leaf. So there's our leaf, our root, and our stem. And now what we can look at in more detail would be those tissues, xylem and phloem. So the structure of xylem is made up of xylem vessel elements, and remember it is there for water transport. Okay, so what the xylem has is it has these quite thick walls where normally a cell wall would be made of cellulose. This cell wall is made of lignin, and lignin is a very, very hard water insoluble product, so it doesn't allow water through, and it's very, very strong for structure, and it can hold things up very, very well. But sitting inside the lignin, you have all of these little things that we call pits, which have cellulose in them to allow the water to diffuse through. So most of it is then going to be lignin. That lignin can be in patterns or it can be solid with just pits. It depends on the plant. But all xylem vessel elements will have lignified walls. Okay, and we can see here where the cells normally would have joined one on top of the other, this area over here, there is no more walls. So we say there is no cross walls. Okay, they have fallen away, so you make one long uninterrupted tube for the water to move down. And now because of this lignin that is impermeable to water and doesn't allow much in or out, all of these xylem vessel elements are hollow because these cells are actually dead. There is no cytoplasm, no organelles, nothing. It is just basically an empty long tube, just like a straw, okay? Because that's what we want for water to move up and down. And that's how xylem is adapted to its function. So then if we look at the phloem structure, so phloem is there for sugar transport. Okay, and it is made of sieve tube elements and companion cells. So we have our sieve tube elements, which are the main parts that will travel with our sugar in it, but then all the organelles that a cell needs are put into the companion cell. So there is my sieve tube on that side, and there is my companion cell. So now these walls are made of cellulose, just like a normal cell. So these are living. So there is cytoplasm inside. Okay, and as you can see, compared to here where there was no walls, here we have a cross wall. And the cross wall we call a sieve plate. So because it's in a sieve tube element, that becomes a sieve plate. And all the little gaps that are in there, we call sieve pores to allow the sugar to move through. So in the companion cell, we are going to have a nucleus, mitochondria, ER, Golgi apparatus, vacuoles, all the organelles that we normally would find in cells are going to be in this companion cell. And then it has connections moving through into the sieve tube element, and those we call the plasmodesmata to allow for easy movement between them, because obviously these need to communicate with each other because they are connected. So the only things that you would find in your sieve tube are very limited organelles. You might have mitochondria because it needs some energy, okay, that you would find in there. Other than that, all of the organelles that it needs are in that companion cell. Okay, so obviously the main difference, where these ones are dead, these ones are living and filled with cytoplasm, whereas those are hollow, okay, and then you can compare both of them for their different functions that we have. Okay, so that is our main plant structures for transport, our xylem and our phloem. So now what we need to look at is how water gets into the xylem and how it moves, and then how sugars get into the phloem and how they move. So if we start with water transport. Water transport is going to start by water needing to move into the root. Okay, so now for water to get into the root, it needs to go from that soil, through that root hair, and all the way into the xylem. And there are two different pathways that water can take to get there. Okay, so we can have water moving through the cells, okay, on the inside, going in, and then moving into it through the cytoplasm, through all the cells in there, from cell to cell, until it gets into the xylem. And that would be what we call the symplastic pathway. So now the symplastic pathway, it moves in and through from cell to cell by osmosis. Okay, so we go water moving in, and because there's a whole bunch of water now in there, it increases the water potential. So the water potential is higher in that cell than it is in that one. So then water moves from where it's high to where it's low. Then it increases the water potential of that one, which is then higher than this one. So water moves from where it's high to where it's low, all the way until it gets into our xylem vessel. So that's the symplastic pathway, moving through from cell to cell through the cytoplasm by osmosis. And then the other pathway is, instead of going through each cell, which can take time to get through, water can move just into the cell walls. And it can go along each cell wall, from cell wall to cell wall, all the way through until it gets to a point. So now moving from cell wall to cell wall is called the apoplastic pathway. Okay, and that happens because our cell wall is made of cellulose, so when water moves into there, it becomes wet, compared to the other one, which is drier with less concentration of water, so it can move from where there's a lot of water to where it's less, and basically diffuse through the cellulose of the cell walls, okay, which is much quicker than going through the cytoplasm slowly, so it ends up getting there very quickly. But at that point where we have our endodermis, it's going to stop because the xylem, remember, has lignified walls. So you cannot diffuse the water through the lignin of the walls. It has to move in via the pits. So to actually get it into a cell, then you have these solid areas inside our endodermis, and that is called a Casparian strip. And the Casparian strip is made of suberin. And the suberin is impermeable to water. So what happens is the water hits that Casparian strip and then it gets forced inside. As soon as it hits there, it gets forced to move inside, and then once it's inside, it can then diffuse into the xylem vessel. So in the apoplastic pathway, it's going to travel along the cell walls until it reaches the Casparian strips, where it's forced inside, and then it will travel to inside the xylem. Okay, so that's the two ways that water get into our xylem from our root, through the apoplastic and the symplastic pathway. So now once it's in the xylem, it now needs to get from those roots up to the leaves where it needs to be used for photosynthesis. So the two ways the three ways in which water moves up the stem is because of forces of adhesion and cohesion that we have, root pressure, and transpiration. So adhesion and cohesion are your properties of water. So adhesion is the ability of water molecules sticking to other things. Okay, and cohesion is water molecules sticking to themselves. So because the water molecules are going to stick to themselves as well as stick to the edges, it means that water is not just going to fall back down, because remember, it's going to be moving against gravity. So because each time it moves up, it is going to stick, it is going to mean that it can then slowly creep its way up. The root pressure is because as the water is moving into the xylem, it's going to keep coming in. So all of that water moving into the xylem has to go somewhere. So it starts to get pushed upwards because of that incoming pressure of water constantly moving in. So root pressure pushes the water upwards to a point. Okay, so if it's a very small plant, it's going to be able to move quite far up. But if it's a very huge tree, your root pressure is going to move up, but then you're going to need something else to get the water to actually move all the way to the top. And that's where transpiration comes. So transpiration, the definition of it is that it is the loss of water vapor from the aerial parts of the plant. So what that means, it's the loss of the water vapor from the leaves. And as water vapor is getting lost, because of the cohesion of the water molecules sticking to one another, as one water molecule evaporates out of the leaf, it pulls the next one and the next one. And you end up with what we call transpirational pull, because it's pulling one molecule to the next one to the next one. So if I have, for example, a string of something all attached to each other, okay, and I think if that is like my xylem, as I pull the one through transpiration, because they're all attached by that cohesion, one of them moving is pulling all the rest. So that's how our water is going to move up by transpiration. Each time one molecule evaporates, it pulls the next one with it. And that's how water moves up our stem against gravity from the roots. And then other factors that can affect that rate, meaning how fast the water is going to move up or down, would be these 4 factors, okay, to affect our rate of transpiration. So the first one is humidity. So humidity is how much water vapor is in the air, okay? So if the humidity is high, that means that if I have my leaf area, okay, so there's my inside and my outside, high humidity means a lot of water vapor all sitting in the air. So if there's a whole bunch of water molecules in there, they don't need to move out because there's no gradient for them anymore. So high humidity decreases the gradient. So that means transpiration is slower. So we decrease our transpiration rate. Okay, but then the opposite thing would happen if it's very dry. If it's very dry, then there's no water vapor here. So then the water would want to move out a lot. Because remember, water molecules, when they're in their vapor form, okay, because it's gases are going to diffuse from where there's high to where there's low until it's even. Okay, so then temperature, if I increase the temperature and it becomes hotter, as I'm increasing the temperature, I am giving these molecules more energy to move. So because they have more energy to move, they are going to move out quicker. So an increasing temperature increases your kinetic energy of the molecules. which means that then they diffuse faster down the gradient. So it would then increase in transpiration rate. And then obviously if I decrease the temperature, it would be colder, so there's less kinetic energy, so then it would be slower. The wind speed, okay, just like that, where I have a whole bunch of water vapor molecules as they're diffusing, if there's a lot of wind, so if I increase in wind speed, it means I'm moving those molecules away. As soon as they diffuse out, I've blown them away. So what I do is I keep getting a gradient there to keep moving. So an increase in wind speed would increase transpiration rate because I'm increasing the gradient. Because I keep moving it away. So I keep having a high amount inside and a low amount outside. And then light intensity, the brighter the light intensity, the more photosynthesis can happen. So if my light intensity increases, it means more photosynthesis. And with more photosynthesis, it means more stomata are open. And if there's more stomata open, there are more gaps for diffusion out, which means I increase in my transpiration rate. Okay, so those are just different factors of how transpiration can change. But now remember, a plant doesn't necessarily always want to lose water, because if it's losing water all the time and there's not a lot of water available, like during drought or in desert areas, then it doesn't want to be losing water. It wants to conserve water. Okay, and then we're going to have other factors that would come into play that would close the stomata to decrease these certain things so that a plant can conserve water. But these are just the general factors that would affect your transpiration rate and how. Okay, so that is basically everything to do with water movement. So then the last thing to look at would be sugars. So now, the process of translocation is the movement of our sugars, okay? And in order to move our sugars, the first thing we have to do is get the sugar into the phloem. And that's done by the process of phloem loading. So, phloem loading is moving the sugars from our general leaf cell, where it's photosynthesizing, and we have sucrose. Okay, because remember, photosynthesis would make glucose, which would join together to make sucrose. Okay, then we have our companion cell as part of our phloem, and then you have the sieve tube, which is where we want the sugar to go. So we want to get it from there all the way into there so that it can move up and down the plant where it's needed. So in order for sucrose to move into the sieve tube, sucrose is a very large molecule, okay, that is a polar molecule, so it can't just move by itself. So what has to happen is in the companion cell, we have a whole bunch of hydrogen ions, okay, and those hydrogen ions are going to be actively moved into the cell membrane space. Okay, so we are going to actively pump them out using ATP for energy, and you're going to actively move hydrogen ions into that intermembrane space so that they are going against their gradient and being pumped out. So there's a whole lot in this space. So that now, since there's a high concentration in the space, we have a plasma, we have a carrier protein sitting in the membrane. So now these hydrogen ions that have been pumped out across the space will want to move back in down their gradient. And when they move back in down the gradient, they take sucrose with it as what we call a co-transporter. So the hydrogen is a co-transporter going back down its gradient and pulling sucrose with it, so that now you get the sucrose in the companion cell, and then it can diffuse through the plasmodesmata into the sieve tube element. So for phloem loading, we have hydrogen ions actively pumped out from the companion cell into the intermembrane space using ATP for energy until we have a high concentration in that space, and it diffuses back through a carrier protein and takes sucrose with it as a co-transporter, and then sucrose moves from the companion cell into the sieve tube via the plasmodesmata, and that's a process of phloem loading. Now there's a whole bunch of sucrose in here, and where there's a lot of sucrose, we call that the source, okay? Where there is lots. And we need to go from a source to a sink. And a sink is where there is very little. And we go from there to there. So to move from the source to the sink, the movement of sugars happens by a process that we call mass flow. Now, mass flow is because now there's a whole bunch of sucrose in there. So now there's a lot of sucrose molecules sitting in there. Sucrose lowers the water potential. And now because I've lowered the water potential, remember sitting directly next to the phloem is the xylem. And now all my water molecules from there, from the xylem, are going to move in to the phloem because it's lowered the water potential. So it goes from where it's high to where it's low. And now the water molecules moving in increase the pressure here. And now there's so much pressure, then it forces everything downwards. So mass flow is where there's an increase in water movement, increasing the pressure, forcing all of the solutions down from their source to their sink, where they would then move out of my sieve tube element to where the sugar needs to be used. So that is our water transport and our sugar transport, and that's all we need to know about plants.